Hi everyone. So welcome to the next lecture on DNA sequencing. So in this lecture I'm going to be covering how you would actually go about analyzing your DNA sequence after next generation sequencing. So after this lecture you should be able to explain how DNA alignment and mapping work in the context of genome analysis and be able to compare and contrast the different kind of processes of doing a de novo versus reference mapped genome sequence assembly, as well as outline the process of conducting and analyzing RNA-seq data. So generally speaking, after doing sequencing, no matter what your kind of goal is for your sequencing experiment, you will inevitably have to align DNA to each other, either to DNA that you've made previously or to some kind of reference. So your goal could be to identify mutations or variants or compare phylogenetic relationships between different organisms to see how closely related your sequence data is to something else that's been published. Or it could be to build longer DNA sequences from shorter ones. So if, say, your goal was to sequence the genome of a new organism, you need to kind of build up all of these reads on each other to work out what the longer DNA sequence is. So all of these things involve alignment. Generally speaking, you're going to have something that's referred to as a reference sequence during this alignment, because you need to have something to align your sequences to, unless your goal is just to align your own reads to each other. This is something that you do in the absence of a DNA sequence, basically where you want to make a new reference, and it's something called de novo assembly, so Latin meaning from new. Essentially, you're not using a reference of any kind, anything that's been published or determined previously. You're just using your own reads to align to each other and see basically how big, how much of a longer DNA sequence you can get from it. So just to show you what I mean a little bit visually, this is our good old DNA sequencer here, a MiSeq, and this is going to give you a bunch of reads. Now, we're going to assume these are all from an Illumina method, so these could all be relatively short, maybe around 125, 150 nucleotides long. And so there's two different ways we can go. We can go to an alignment-based method. So in green, we've got our reference genome, and you can see that some of our reads are aligning to various places on these genomes. So basically we're getting the same sequence in our reads compared to the reference, and so they will map to that region. So some of these are kind of overlapping. There's some parts which are kind of present in one read but not in another read, but then there's parts that are overlapping in all of these reads. And in some cases you might get reads which completely overlap and are basically kind of duplicates from one area of the genome. And this can occur in mapping for different reasons that I'll go through later. Then you've got your de novo assembly method. So we don't have a reference, we're trying to make a new one. We are basically just trying to find overlaps in your own reads and make a new assembly or make a new reference. And so this is a new sequence, which is probably not going to be complete, but it may be new DNA information. So I'm going to talk about kind of alignment and first, and then I'll talk about de novo assembly a little bit later on. So how do we do alignment? Now, I've kind of broken it down to a much shorter or a much simpler example here. Let's say, for instance, this is our reference. This is just a chunk out of a much larger sequence, but could be from the human genome, so something quite long, about 3 billion base pairs. And here is our short read. So you can see here that there are some similarities between our read and the reference, especially if we change the reference slightly or change how our read maps to it. So you can see here there's a match between these two T's and these two T's and these two C's and these two C's, but in order to have them align nicely, we might want to insert a gap into our reference, and that would allow us to get these perfect matches in between these 5 nucleotides and this A over here, but then we have a gap here for this G, and then we have 2 mismatches for these 2 Gs. So this is generally speaking how sequence alignment works, but the complication is that sometimes we have reads that can map to different parts of the genome, especially if we allow these kind of gaps here in the reference to allow our read to map to a certain area. It may mean that that read maps to different areas. And so the way that we kind of solve these issues is by giving a score to the best match for how a particular read matches or maps to a particular part of the reference. So we'll go through exactly how that works. Essentially, this is a simplified way of doing it. This is a matching table, but if we have our reference here, or one sequence and then another sequence, whenever we have a match between the first sequence and the second one, we give that a positive score. So if we give that a 10, that's our kind of running total of 10 for this match, and then we have a mismatch, well, that gets penalized. We give a -5 for that, and that -5 basically takes away from the 10 that we had before. So we have a cumulative score running over the length of this this alignment. Now after that first mismatch, we had another mismatch, so that's another 5 there, and then another -1. So in some alignment cases, it gives you a lower penalty for cumulative kind of gaps. And then we have another 2 matches here. So when you add up all of the positive points and subtract the negative penalties, it gives you a cumulative score of 19. So this is basically just a simplified example of how alignment scoring works. And bioinformatics is perfect for this kind of job because it's very easy to write computer programs that will essentially do this automatically for your DNA. However, computer programs, as great as they are, do run into some problems, especially when there's multiple ways of doing the same thing. So here we have another example. Here we have perfect matches for these 3 A's. We have perfect matches for this GAA, so that's probably going to mean that the computer is going to try to align this read to this reference, but there's different ways that this could be done. So we could insert a gap here, which means we have an insertion of this T in our reference. We could delete an A and a G here as well, and then have a substitution of 2 more nucleotides. That would be one way of doing that alignment. We could instead delete a C from over here and then have 2 substitutions. We could have a number of substitutions or SNPs and then 1 deletion, or we could have a complete deletion and complete insertion. All of these alignments can be done, and they all end up giving different alignment scores. So I'm not going to tell you which of these alignments is correct, because there isn't a correct way to do this. It's just important for you to be aware that when you're doing this kind of analysis, that depending on the tool you use, they will do this in different ways, and it's important to be aware of these differences. However, once you've actually done your alignment, what do you end up with? So this is an example of how we would visualize alignment of our sequence data to a reference. So we have here a chunk of a larger genome. So this is the APL3 gene. So this is showing you that we have a protein coding gene here. These are likely to be some introns and exons. You can see there's kind of blocks and lines connecting them. That usually means that there are protein coding regions, and then they're kind of interspersed with non-protein coding regions of a gene. And then we have these 2 parts down here. So in the middle, we have something showing the coverage. This is basically how many reads have mapped to each part of this genome, and that's also represented by these color-coded reads down here. So we've got these red and blue boxes, which are representing reads from each strand of the DNA, which have mapped to this region of the genome. And you can see here that the coverage graph basically shows you how these reads have kind of stacked on top of each other in these different parts of the genome. You can see there's a little bit of a dip here in the coverage. Basically, that's showing you how that this particular part of the genome, less reads have mapped to it. So if you were analyzing this genome, you might treat this part of the genome a little bit differently, simply because you have got less data telling you that this is definitely the DNA sequence in that location. You can also see that we've got these kind of indicators here, these lines going through the data. These could basically be showing you mutations or SNPs in your reads relative to the reference. So they're showing you places where your sequence are different to the reference. The reads have still mapped to this location, but it's showing you what could be a key difference in your sample relative to the reference. Now, in order to improve or kind of overcome some of these difficulties when it comes to aligning and mapping your reads to the reference genome, what's often done with Illumina and the kind of short sequence-based next generation sequence technologies is something called paired end sequencing. Basically, in my previous lecture, I talked about how Illumina data or Illumina DNA samples needed to be fragmented. So here we have a fragment of DNA here. What we can actually do is we can pair reads to either end of these DNA fragments and have a known distance in between the ends of those two reads. So these reads here might be the usual 125 or 150 base pairs for Illumina, and then there could be what's known as an insertion or an insert of about 700 base pairs, which separates them. So when it comes to your data analysis, when it comes to mapping these reads, you know that read 1 is associated with read 2 and that there should be a distance of around 700 base pairs in between them. So when you map these paired end reads to your reference, you'll see that read 1 is over here and read 2 is over here, and they will map to the reference, or should map to the reference in this way. The benefit of this method is the fact that if you have unexpected shorter or longer distances in between your paired end reads, this can help kind of solve issues when it comes to mapping, particularly when it comes to repeats and deletions. Now, repeat sequences in this case basically refers to the same DNA sequence being present in your reference in two different locations. So we've got a repeat present here, and if you didn't have this paired end read, you wouldn't know whether or not this read mapped to this location or this location. But if you know that this read is paired to this read here and that there's a distance of 700 base pairs between them, you know that this read should map to this location rather than the other. So this can be really helpful for solving where your reads are supposed to map to in repeat regions, or when you have insertions or deletions that cause your paired end reads to map to places that should be a bit closer together. So these are deletions in your kind of sample relative to the reference. So basically you can use the paired end reads to put together your kind of contigs into scaffolds, because even if you have a gap in between your reads and you don't know what the sequence is between them, if you assemble your reads into contigs, so you assemble your reads into longer DNA sequences, and you have one read from a paired end read that's at one end of one contig and the other paired end read at the other end of another contig, you know that there is a relationship between these two contigs, and you know that they should be the order that they should be in the genome. So when you put together your kind of higher order scaffold of the genome, it can help you determine how these contigs are arranged, which is very, very helpful when it comes to doing a genome assembly kind of experiment. So to move on to the other type of assembly here, de novo assembly. As I mentioned before, this is just basically putting together DNA sequences from your reads alone. In order to do this, you're still going to be using the same kind of alignment-based principle, so you still need to align your sequences, so you're still going to be using this scoring method, but you have no ability to map to a reference. And so what you're going to be doing instead is, or what the computer, your bioinformatic algorithm, is going to do, is try to build a graph of how each of these reads are connected. So where you have a little bit of overlap between these reads, it can kind of build a contig, or it can try to put together a kind of network of these reads based on sequence overlaps. This is a computationally pretty intensive program because of the number of sequences that are involved, anywhere from the thousands to tens of thousands. Some parts of the genome, so our repeat sequences, aren't really sequenced very easily, so this adds to the difficulty. And again, if you're using sequences that have a higher error rate, so the PacBio and Nanopore do have higher kind of error rates, then this is going to affect how well your reads are going to align to each other. Essentially, this is the kind of process. So here are our reads. They're all kind of color-coded, and the colors are showing you the sequence similarity. So these orange parts here should represent the same DNA sequence, so they're going to align together. The purple parts are representing the same sequence. So when you try to identify these overlaps, this is basically what you're going to get. You're going to get a series of reads that overlap with each other. What you can do then is collapse those overlaps down to contigs, where you build a larger series of sequences based on those overlaps. So you're connecting kind of the dots in between each of these reads to make a longer sequence. Then there's a kind of more kind of complex computational process involved, which can use paired-end sequencing, where you're basically trying to find connections in between those contigs, and in the end you'll end up with your consensus sequence, which will have kind of gaps in it. So this is complex, but still the kind of simplified visualization of how this would occur. In reality, it's even messier, because you get these kind of graphs being constructed by your computer, and this is essentially what it's trying to do on your behalf. Now, in terms of giving some reliability to your de novo sequence assembly, there's a really important metric which is often reported called the N50. So this is a number, basically a single number, which determines how long most of your contigs are. So the N50 is the length of the contig in which 50% of your DNA are basically in it. So just as an example, if we have 7 contigs, we have 2 contigs that only have 1 base, and then a few that are a bit longer, going from 3 to 20. You sum up the total number of bases that you have, or nucleotides that you have, so you have 50. 50 is the total, so the N50, or the halfway of 50, is going to be 25, and so you would basically choose the contig that has at least 25 bases within it. So if you choose your N50 of 12, then all of these, the sum of all of these smaller numbers adds up to 30, which is higher than 25, so your N50 is going to be 12. Finally, just in terms of genome assembly, this is really quite a common kind of practice in DNA analysis these days. However, most genomes aren't actually sequenced to completion. Most DNA assemblies, or most genome projects, are sitting at the draft level, where you have a number of these non-linked scaffolds and you don't know how they're arranged or what the gaps are between them. But on the other hand, they're pretty easy to get, because you can get them from a single sequencing experiment. If it's done fairly well, hopefully you won't have too many scaffolds, maybe half a dozen or so. but it might be very difficult to close those gaps, and that's basically what a closed or finished DNA sequence is, when you have a single contiguous DNA sequence for each chromosome. Generally speaking, we can do it for small genomes like the ones in viruses and bacteria pretty easily if you're combining short read technologies like Illumina with long read technologies like Nanopore and PacBio. However, if you're trying to do this for eukaryotes which have large genomes, then you generally need collaboration between quite a few labs. Finally, the last thing I want to talk about in terms of analysis is RNA. So it's actually quite easy to modify next generation sequencing technology to analyze RNA instead of DNA, because all you have to do is essentially isolate RNA from your sample instead of DNA and then reverse transcribe it. So you can do this simpler by using reverse transcriptase. You convert your RNA to cDNA. If you're doing an Illumina-based method, which usually for RNA-seq you would be using, you would fragment it, do your next-gen sequencing, and then align your sequences to the reference genome. So if you have more sequences that map to particular parts of the genome, then it means that those parts of the genome are more expressed. So these could be genes, or they could be highly expressed non-protein coding regions like tRNAs or something like that. Some technologies, basically the nanopore, can actually sequence RNA directly as well. RNAs can be fed through the nanopore, and it will give you reliable sequence data from that. So this is the kind of simplified visualization of how that would occur. You've got some mRNA. This is from a eukaryote, so it's got a poly A tail. You can fragment your RNA or reverse transcribe it to cDNA, add your adapters, so this is for an Illumina-based run, obtain your sequence information after next-gen sequencing. Then you've got your reads. You map your reads back to your reference genome, so basically the sample that you obtained the RNA from in the first place, and then have a look at the relative expression level at that part of the genome based on how many reads map to it. So you can see here that there's kind of a higher amount of reads in this region, and a slightly lower amount basically corresponds to a decreased RNA expression level. RNA-seq is pretty useful in terms of measuring the amount of RNA being expressed by genes. So it's very useful for quantification or measuring how much a particular gene is expressed. Is it expressed a lot or not very much? And it can do this in absolute terms, so giving you a number of the number of RNA molecules. Or what's also done commonly is comparing RNA between different samples, and it will just give you relative RNA expression between, say, a treatment and a control sample. The other thing that RNA-seq is very good at is actually telling you about new RNA species, because unlike microarrays, you don't need to have a probe designed for an RNA in order to detect it. It can detect all of them without any prior knowledge. So it can tell you about RNA molecules that are kind of alternative splice variants of eukaryotic genes, so new ways that introns are spliced, or exons are spliced together in eukaryotes, novel genes that haven't been annotated previously, different sequence variants of genes, whether they're kind of edited at the RNA level, and also the location and expression of non-coding RNAs. So just to summarize kind of this whole lecture, here's an image that shows different ways that next generation sequencing can be analyzed at either the DNA or the RNA level. So depending on whether or not you wanted to do some targeted sequencing, so this is basically sequencing your sample versus reference, sequence, map to a reference genome, detect new SNPs, indels, copy number variants, etc. You might only just be interested in exomes rather than the non-coding parts of the genome. So this is a kind of specific type of targeted sequencing where you're just sequencing those exomes. But generally speaking, it's still going to be a similar process where you're sequencing the DNA and then assembling it together. You might be interested in metagenomic sequencing, so sequencing from environmental DNA, which generally tends to involve de novo assembly and annotation. You might be interested in epigenetics. There's different ways that we can modify our protocol to focus on histone modification or DNA methylation. And of course, for RNA, there's a number of different ways we can utilize next generation sequencing to examine RNA, either for quantification or for identification of different or new RNA species. So if you'd like to read about these types of analysis in more detail, the best place I've found to go to is this Use Galaxy website. It has some really great tutorials, not only teaching you about the theoretical basis, but actually how to do this in practice with current open source bioinformatic tools. Otherwise, I hope you enjoy these lectures, and I'll see you again soon.